The human genome is a complete set of nucleic acid sequences for humans, encoded as DNA within the 23 chromosome pairs in cell nuclei and in a small DNA molecule found within individual mitochondria. These are usually treated separately as the nuclear genome and the mitochondrial genome. Human genomes include both protein-coding DNA sequences and various types of DNA that does not encode proteins. The latter is a diverse category that includes DNA coding for non-translated RNA, such as that for ribosomal RNA, transfer RNA, ribozymes, small nuclear RNA, and several types of regulatory RNA. The human genome is contained within the nucleus of cells. The genome is well protected, but many things can cause DNA damage, including aging, some drugs, and environmental factors like UV rays or pollution. DNA damage is defined as any modification of DNA that changes its coding properties or normal function in transcription or replication. DNA damage is an abnormal chemical structure in DNA, while a mutation is a change in the sequence of base pairs. DNA damage contributes to the mechanisms of aging and disease. It has broad relevance to human pathobiology, with its involvement in birth defects, cancer, premature aging syndromes, and certain neurologic disorders. DNA damage can be a major problem, and the cell has mechanisms to detect and repair that damage. When both strands of a DNA molecule are broken, it's known as a double-strand break DSB, and scientists have now revealed new details about how these problems are fixed. This study, which has been reported in Nature Structural and Molecular Biology, has shown that after a DSB, the nucleus is actually prodded by parts of the cellular architecture or cytoskeleton, called microtubules, which are in the cell cytoplasm. These microtubules also interact with the nucleus of the cell to capture and repair DSB. Think of it as the nuclear envelope, growing long finger-like structures that help tie pieces of broken DNA together, akin to tying our shoelaces. In this study, the researchers treated cells with chemotherapy drugs to trigger double-strand breaks in the nuclear DNA, and super-resolution microscopy was used to create 3D models of the tubule network. Microtubules in the cytoplasm poke the nucleus, causing nuclear envelope tubules to form inside of the nucleus and attract DSB. Computational models indicated that a network of nuclear envelope tubules forms throughout the nucleus and contacts non-peripheral DSB. The DSB capturing nuclear envelope tubules that are created are supported by molecular complexes such as the linker of nucleoskeleton and cytoskeleton complex link, nuclear pole complex NPC, and nuclear actin filaments. The survival of cells and DNA damage repair relies on this process. Researchers also investigated BRCA1-linked breast cancer to learn more about how these tubules affect the growth of cancer cells and their impact on cancer treatments. There are usually extremely high levels of DNA damage in cancer cells, making them particularly dependent on this mechanism for growth and survival. Patients with cancers that are difficult to cure need novel therapeutic options. This is the case of women with aggressive types of breast cancer. This study demonstrates that targeting double-strand break capturing nuclear envelope tubules represents a great therapeutic opportunity to kill these aggressive cancers. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.